Hello everyone, this is Dr. Leili Hatami. Welcome to the Human Anatomy course, Chapter 2, Cells, the Living Units, Part 3, Nucleus. In Part 1 and Part 2 of Chapter 2, we studied the plasma membrane and cytoplasm as two main components of the cell, and in this video I'm going to cover the nucleus. The nucleus is the control center of the cell. It's directing all the cell's activity. Inside the nucleus, you find the genetic material, which is called DNA. DNA provides instruction for making proteins. Nucleus is approximately 5 micrometer in diameter, and most cells have one nucleus. There are some cells like skeletal muscle cells which they have many nuclei. These cells are so long, and then you can find many nuclei under their plasma membrane. And also there is a cell in our body which does not have any nuclei. It's mature red blood cell. Mature red blood cell is anucleated, which means that does not have a nucleus. The A means no. The nucleus has three main parts: the nucleolar envelope, nucleolus, the genetic material, which is chromatin, and chromosome. In this picture of the nucleus, you can see how the nuclear envelope is going to cover the nucleus. And as you can see, it has two layers of membrane, which between these two layers, there is a fluid which gonna fill this space. And then in the center of the nucleus, there is a tiny structure. It's called nucleolus or little nucleus. And also, if I go back to the nuclear envelope, you can see that across the nuclear envelope, there are some little openings which allow the molecule, the certain molecule, pass in and out of the nucleus, from the nucleus to the cytoplasm, or from the cytoplasm to the nucleus. And then inside of the nucleus, we will find the genetic material as the chromosome or chromatin. Okay, let's go and find these parts of the nucleus one more time. The nuclear envelope is the protective barrier surrounding the nucleus, made up of two parallel membranes separated by fluid-filled space, and helps to keep the cell's genetic material, DNA, safe and organized. And you saw that at the nuclear envelope, there are some little openings, which is called nuclear pores, which the nuclear pores allow certain molecules to pass in and out of the nucleus. These nuclear pores are crucial because they enable large molecules like RNA and also proteins involved in the cell processes to move between the nucleus and the cytoplasm. The other structure related to the nucleus or part of the nucleus is nucleolus or little nucleus. It's a tiny structure situated in the center of the nucleus. The nucleolus contains parts of several chromosomes, and it's small but is crucial part of the nucleus because it's the place where the cell builds its ribosome. We learned about the ribosome in part 2 of chapter 2. Ribosome are the protein synthesis factory, but guess what? The ribosome, it's gonna be made by the nucleolus. Next, let's go over the genetic material of the nucleus, which can be called chromatin or chromosome. Chromatin is the cell's genetic material. It's made up of DNA, which carries the cell's genetic instructions. In a cell, DNA does not usually exist by itself, but instead associates with specialized proteins that organize it and give it structure. These proteins are called histone proteins. So simply means that chromatin is composed of DNA and histone proteins. Consider chromatin in two states. We have condensed chromatin and extended chromatin. Condensed chromatin, or inactive form, it is tightly coiled strand of DNA. In this state, the genetic instructions are sort of packed away, not actively being used. But the extended chromatin or active form, it is uncoiled strand of DNA. In this state, the genetic instructions are ready to be accessed and used by the cell. Okay, or let's do this. Think of chromatin as the cell's instruction manual. When the cell needs to make proteins, it uncoils the relevant part of the chromatin. So we can have the extended chromatin. It copies the genetic code onto mRNA and starts building the protein. Okay, so it means that the most active cells have a lot of extended chromatin, meaning they are actively using the genetic instructions. For example, the tumor cells, which they are actively dividing and producing the protein, or the embryonic cells also, they are so active, and we can find large amount of extended chromatin and little condensed chromatin in their genetic material. But on the flip side, cells with condensed chromatin might be in sort of standby mode, not actively using those genetic instructions at the moment. Okay, but what is the chromosome, or what is the difference between chromatin and chromosome? Chromosomes are the well-organized, compact version of chromatin, especially during the cell division. Each chromosome is composed of a single long molecule of DNA, and each human cell, like the typical human cell, has 46 chromosomes, or 23 pairs of chromosomes. Let's look at the DNA structure. Okay, this is a DNA structure which you can see is like a twisted ladder made up of pairs of the nucleotides, which the nucleotides are the basic units containing the information that guides the development and functioning of living organisms. Nucleotides are composed of the nitrogenous base, which they can be adenine, thymine, cytosine, and guanine, A, T, C, and G, plus a pentose sugar, which is deoxyribose sugar, and a phosphate group. In this image, you can see a chromatin and chromosome structure. Before replication, before the mitosis, before the cell division, DNA exists as chromatin. DNA condenses and coils into the X-shape form of chromosome. So it means that in this part, you can see the chromatin, and now the chromatin just condensed, and then you can see this X-shaped form of a chromosome. Only after the DNA has replicated, you can find this picture or this shape of the genetic material. Because DNA has already replicated, each chromosome actually consists of two identical copies. The two copies are called sister chromatid. So each of these is called the sister chromatid, which they are attached to one another at a region called this centromere. So here, this button-shaped structure is called the centromere, which the centromere is going to connect these two sister chromatids to each other. Okay, let's go back and look at the chromatin structure one more time. So as you can see, chromatin is composed of the DNA molecule that you can see here, and then the histone molecules, and you can find in some areas we have the extended chromatin, the uncoiled strand of DNA, which means that it's active and it's gonna be under like transcription and then or copying, and then the next part that you can see is very condensed is the inactive part or the inactive form of the chromatin, which means that it's not actively being used. Okay, so far we talked about the nucleus, which is the control center of the cell, the genetic material which is running all the activities in the cell for the living organism going to be found in the nucleus. We talked about the nuclear envelope, the nuclear pores, the nucleolus, what is the nucleolus function or job, and then a little bit we discussed about the genetic material, structure and form as chromatin, chromosome, DNA, and now we are going to talk about the cell life cycle. The cell cycle is the series of changes a cell goes through from its formation to its reproduction. It's like the life journey of a cell from its creation to the point where it splits into two new cells. So as you can see in this picture, it's showing the cell life cycle, that the cell, which you can see this one cell, it's going through some processes, and then we can divide it to two new cells. We can divide the cell life cycle into two major periods, interphase and mitotic phase. The interphase is like a preparation phase where the cell gets ready for division, which division is happening during the mitotic phase. So if you look at this shape, you can see that this like a blue color, it shows the interphase. So as you can see, it's a kind of long phase. Around 90% of cell life gonna be in the interphase. And then we have the mitotic phase, which is the cell division phase. And then during the mitosis, the cell splits into two new cells. So as you can see, the interphase is before mitotic phase, which is kind of making the cell ready for division. Okay, let's study the interphase, and guess what should happen during the interphase? Obviously, it is a time when the cell is actively doing its day-to-day activities, but at the same time it's going to get ready to divide. Okay, so for the mitosis or the cell division, we will see that a mother cell or a parent cell, just for example, it has 1, 2, 3, 4 chromosomes. During the mitosis or after the cell division, it splits into two cells, the exact size, the exact shape, and the exact number of chromosomes. Okay, so that's the reason we have to make ourselves ready, because after cell division, we want to have the same shape, the same size, and the same number of chromosomes. So it means that before cell division, during the interphase, the cell must grow, right? The cell must grow, and also it must copy the genetic material, the DNA. Then we're going to have two exact shape and size and chromosome number like their mother cell. So then the interphase is the phase that cells get ready to divide. The cell must complete several important tasks, like it must grow and copy its genetic material. The interphase, it has three substages or three subphases: G1, S, and G2. G stands for growth. In some other textbook, it calls this as gap. So we have growth 1 or gap 1 phase. S stands for synthesis, S subphase, and then G2, which means growth 2 subphase or gap 2 subphase. Let's see what is happening during these subphases. G1 phase or growth 1 is the first part of interphase, where the cell is actively growing, carrying out its usual activities. Okay, so then at the end of the G1 centrioles, which we talked about the centrioles before, it's one of our cytoskeleton, which is an important tool for the cell division. It began to replicate. We're going to have a copy of the centriole near the end of G1. Next phase is S phase or synthetic phase. It's time when the cell's DNA copies itself to ensure that when the cell eventually divides, each new cell will have an exact copy of the genetic information. So it means that during the S phase, we have DNA replication or copying of DNA. And we know why the synthesis of DNA should happen. Because we want each of daughter cell at the end receive the identical copies of genetic material like their mother cell. Okay, and then let's go to the final part of the interphase, G2 or growth 2. It's like the cell's last minute preparations before the big event, which is division. It's the final stage of interphase. During the second gap phase or growth phase, cell is still growing and making proteins and organelles. Centrioles finish copying themselves at the end of G2, and then we can say by the end of G2 the cell is ready to divide. Okay, so now let's review the interphase one more time. The prefix inter means between. It's reflecting that interphase takes place between one mitotic phase and the next one. If I go back to this picture, you can see that the interphase, it means the phase between the one mitotic phase and the another one. Okay, which we divided to the G1, S, G2. We know during the interphase, the cell is growing, protein gonna be copied, the organelles gonna be copied, and also during the S phase, DNA gonna be copied as well. Now our cell is completely ready for the cell division. Okay, now let me ask you, why we need the cell division? Why cell division is essential for our body? Consider the example of 2-year-old cute baby version of yourself and compare it with your current appearance. Notice the changes. The first thing that you can see is you got taller. Your height increased, right? That's because of mitosis. Throughout your life, you may have experienced cuts on your skin. However, if you inspect those areas now, you'll likely find that cuts have disappeared. It's because of mitosis. Mitosis plays a crucial role in replacing damaged or dead cells. Cells in our body have different lifespans. For instance, some last only a few hours, like certain white blood cells, while others can be alive for days or even months, such as epithelial cells. So we can say that through mitosis, we can replace the new cells. we can replace the damaged cell or the dead cells by the new cells. Okay, so in general, mitosis is essential for growth, as we all experienced it. Mitosis is essential for tissue repair. Also, we experience this as well, and also for replacement of the cells that have reached the end of their lifespan. It's a dynamic process that ensures our body maintains its structure and functionality over time. Cell division involves mitosis and cytokinesis. The mitosis is simply means division of the nucleus. So during the cell division, nucleus is dividing to make sure each daughter cell or daughter nuclei is going to receive the equal amount of chromosome as their mother cell. And cytokinesis means division of the cytoplasm, which occurs like at the end of the mitosis or kind of after the nucleus divides. So make sure you know the mitosis is division of the nucleus and cytokinesis is the division of the cytoplasm. Mitosis, which is division of the nucleus, it has 4 stages. We can use this mnemonic PMAT for the stage of mitosis in order. P stands for prophase, which is the first and longest phase of mitosis. The second phase is metaphase. The third phase, which is so short, is anaphase, and the end of the mitosis is telophase. But don't get too hung up on names. What's most important to understand is what is happening at each stage, which now we are going to focus on each stage event, what is happening during prophase, metaphase, anaphase, and then finally telophase. Prophase is the first and longest stage of mitosis that we can divide the prophase to early prophase and the late prophase. In early prophase, the genetic material chromatin start to condense, and then we call this as chromosome, making them easier to pull apart later on. Each chromosome is a pair of identical threads, and each thread is called a chromatid. We have 2 threads called sister chromatid, which, as I mentioned before, they are held together by this button-shaped structure, as it called centromere. And also the centriole pairs, which we copied them during the interphase, they are separating from one another. And then the mitotic spindle began to form. The spindle is a structure made up of microtubules, the strong fibers that are part of the cell's skeleton. Its job is to organize the chromosomes and move them around during the mitosis. The spindle grows between the centrosomes as they move apart. During the late prophase, the centriole is still continuing moving away from each other, and the nuclear membrane fragment. So we can say that in general, during the prophase, the cell starts to break down some structures like the nuclear envelope fragments, and also build others up, like we can build the mitotic spindle, and then we have some other changes like movement of the centrioles. So it means that if I want to draw the early prophase, I can draw something like that. Plasma membrane, the nuclear envelope, then here we will have the chromosome start to condense, then we won't see the nucleolus anymore. So then here also we can find the centriole as these paired structure, the mitotic spindle begins to form between this centrosome or centriole. This is the early prophase event happening. And then for the late prophase, also we can have the plasma membrane, then the nuclear envelope has been fragmented, and now the chromosome has been released. So we can see the chromosome like that, but there is no nuclear envelope around them. And as I mentioned before, the let me use the red color, centrioles moved apart, and now we can find them in two different side of the cell. And then you can see how the mitotic spindle is going to be formed between these centrioles. And then, so as I mentioned, there is no nuclear envelope. The nuclear envelope breaks down, the chromosome fully condensed, and now we have these two centrioles in two different poles of the cell. Okay, next, we have this picture that one more time we can review the cell division. So we can have the interface, which is the phase before cell division or between the mitotic phase and the next mitotic phase. So as you can see here, we can find the nuclear envelope. You can see the nucleolus, then the chromatin, then the centrosome, which now we have two centrioles. And then for the early prophase, as you can see, the chromatin has been condensed, and now we have chromosome. Each consists of two sister chromatid held together by the centromere. Still you can see the nuclear envelope, but the nucleolus has been gone. And then you can see how these centrioles are moving away from each other. Mitotic spindle has been formed, and then at the end of the prophase or late prophase, the nuclear envelope is breaking down, the chromosome has been released, and then we can see the centrioles at two different or two opposite sides of the cell, and then this mitotic spindle also has been grown, and then we have something like that. Okay, what is the next phase of the mitosis? Is metaphase. The metaphase is the second stage of the mitosis. In metaphase, the spindle has captured all the chromosomes and lined them up at the middle of the cell. So again, if I want to draw a cell during the metaphase, there is no nuclear envelope, the chromosomes are released, and now we can find them in the midline of the cell that they are actually going to be held by the spindle, the mitotic spindle, something like that. So just remember the M at the beginning of the metaphase and then the term midline. Now we have chromosome lined up at the midline of the cell. So M for metaphase, and don't forget at this phase, all the chromosome lined up at the midline or middle of the cell. Okay, next phase, which is the anaphase, is the third and shortest stage of the mitosis. The sister chromatids separate from each other and are pulled toward the opposite ends of the cell, or we have like a pulling away chromatids from each other. So again, the A at the beginning of the anaphase and pulling away from each other. So we can say that this is the event happening during the anaphase. So if I want to draw one more time a cell during the anaphase, I should draw the centrioles, the mitotic spindle, something like that between these two. And then we can see that how these chromatids are pulling away from each other toward the opposite side of the opposite pole of the cell. And then in telophase, the cell is nearly done dividing. This is final phase of mitosis. And during this phase, chromosomal movement stops, and then chromosome gonna be found at opposite poles of the cell, and they're starting to being uncoiled. The two new nuclei form, one for each set of chromosome, and the nuclear membranes and nucleoli reappear. So let's look at this picture, and as you can see here, we have the metaphase, all of the chromosome lined up at the midline of the cell. Then during the anaphase, the chromatid are pulling away from each other, and during the telophase, we have the nuclear envelope forming one more time. There is no movement of chromosome anymore. At each pole of this cell, we have the chromosome, the genetic material, the same or number of organelles, so the same number of chromosome. Okay. What's gonna happen during this kind of time? Cytokinesis is happening, which is division of the cytoplasm to form two new cells. The cytokinesis overlaps with the final stages of mitosis. It may start in either anaphase or telophase, depending on the cell, and finishes shortly after telophase. And now we're going to have two daughter cells with the same size, same shape, and the same number of chromosomes as their mother cell. I hope you got the concept of the mitosis or the cell division, which, as we discussed about this, it has prophase, metaphase, anaphase, and telophase. So we copied the DNA and now, or chromosome, and now for each cell we have the same number of chromosomes. The next concept is the cellular diversity. We mentioned before at the beginning of chapter 2, the human body is composed of trillions of cells, and we have more than 200 different types of cells. Their shape are different, their sizes are different, they have different names, they're going to be found in different location of the body, and they have different functions. The specialized function of the cell relates to the shape of the cell and also arrangement of the organelles. We made the example before. For example, a cell which is specialized for producing the protein or lipid, they must have actually the different amount of organelles like rough endoplasmic reticulum, smooth endoplasmic reticulum, or the other organelles. Okay, but let's look at these different cell types with different functions. There are some cells in our body, they're specialized to connect the body parts or cover or line organs. For example, the fibroblast. This is a fibroblast, as you can see, it's a spindle-shaped cell. It is the cells of the connective tissue, and it's specialized to make and secrete the protein components of fibers. Erythrocytes or red blood cell, they're going to be found in our blood, and their shape is like a concave shape. They do not have a nucleus, and they're going to be filled with the hemoglobin, which is specialized to carry the oxygen. And also, as you can see, the erythrocyte shape is it has a concave shape, so it's going to provide the surface area for uptaking the respiratory gases, especially oxygen. And the next cell, which is epithelial cell, it has a hexagonal shape, which it allows the maximum number of epithelial cells to pack together. So as you can see, they're going to be very close to each other, kind of joining to each other. So this is the way that you can see we have like a different shape of cell in different location with different functions. Next group of cells, they specialize to move organs and body parts. We have skeletal and smooth muscle cells. As you can see in this picture, these cells are elongated cells and filled with actin and myosin filament, which the actin and myosin filament are the contractile protein which the cells need to contract and make movement. Next group of cells, they can store nutrients. The best example is fat cell or adipocyte. Inside of these cells, there is a large fat droplet. As you can see, this large fat droplet occupies the most part of the cytoplasm as it's going to push the nucleus at the periphery of the cell, and the other cells can fight disease. These can be counted as our immune cells like macrophages. The macrophage moves through the tissue to reach the infection site, and through the phagocytosis it can digest or destroy the microorganism. And the next cell, which it gathered the information, is nerve cell or neuron, which as you can see is kind of a weird shape. It has the long processes, which is specialized for receiving and transmitting the messages. Or we have some cells, they are reproduction cells or sex cells like sperm. Look at the shape of a sperm cell. It's so unique, right? It has a very long tail, which it allows the sperm to swim to the egg for fertilization. And also it has like many mitochondria in the middle part of the sperm because for the movement it needs a lot of energy. So in this way, we learn that even all the cells has the nucleus, cytoplasm, and plasma membrane in common, but based on their function, they can have different structure, different shape. They can be found in different location of the body with different name. And at the end, there are roots to remember that is helping you to memorize the term which we have in this chapter, like the anaphase, as ana means a part. So just remember that the anaphase is the phase that the chromatin are taking apart or pulling away from each other. Or we had this term bilayer, and we know the bi means two, so bilayer it means we have two layer. Or we had the organelle, means or L means small or small organ or little organelle. Or when you know the phago means eat, pino means drink, so it's kind of easy to remember phagocytosis, pinocytosis, and the other stuff. Okay, thank you so much for watching. If you have any question, feel free to send me an email and have a good day. Bye.